Okay. Okay, so I have received a signals that we will receive our arrival of the guests in 3 minutes time. So now this is a housekeeping announcement. We kindly remind everyone to switch your mobile phone to silent mode to ensure a smooth and uninterrupted session. And now please pay attention for the safety video announcement.
May we have your attention, please? This is a public safety announcement. Assalamu alaikum wa rahmatullahi wa barakatuh.
Welcome to International Islamic University Malaysia, Kuantan campus. You are now in the Grand Hall.
Upon the sound of a fire alarm or when evacuation order has been issued, please evacuate the building immediately. Emergency situations can be triggered by the presence of potential hazards such as fire and natural disaster.
This hall has 9 main emergency exits.
Please take notes on emergency evacuation routes, locations of exit doors and staircases. Exit signs will remain illuminated at all time. Please take a moment to locate the nearest exit to you. During evacuation process, please remain calm and walk to the nearest exit according to predetermined evacuation routes. For faster and safer evacuation process, please refrain from talking and running. Once you are outside the building, please proceed to the nearest assembly point and wait for further instruction. Thank you for your attention. This message is brought to you by IIUM Safety and Health Committee. Safety is as simple as A, B, C. Always be careful.
Bismillahirrahmanirrahim. Ladies and gentlemen, please rise for the arrival of our honorable guests and VVIPs. Announcing the arrival of our distinguished guests. As a mark of respect, we will now begin with the national anthem, Negaraku, followed by the IUM anthem, Leading the Way.
Lighten the future, share the wisdom to the spirit of Islam. Revelation and reason, we shall excel, we shall prevail. Merging faith and knowledge, we, the Khalifa, we will fulfill the Amana. Together we make the world a better place. Forever expand the culture of Ikra. IUM is to realize the meaning of Ramadan lil Alameen. Together we make the world a better place. Forever expand the culture of Ikra. IUM is to realize the meaning of mercy to all worlds for mankind.
Thank you. Please be seated. Assalamu alaikum wa rahmatullahi wa barakatuh, and a very good morning. It is my utmost pleasure to extend a warm welcome to our esteemed guest, Professor Dr. Samsul bin Rahman, members of the IIUM Board of Governors, Associate Professor Dr. Razman bin Mohammed Rose, Deputy Campus Director, Student Development and Support Services, Professor Muhammad Lokman bin Mad Isa, Director, Institute of Planetary Survival for Sustainable Wellbeing, PLANETIIUM, and our distinguished speaker today. Next, Assistant Professor Dr. Rizwanul Hakim bin Abdul Razak, Deputy Director, PLANETIIUM, Assistant Professor Dr. Muhammad Shukri bin Baba, Acting Chairman for Professorial Lecture, Professor Dr. Susanna binti Abdul Rahman, We also extend our warmest greeting to all deans, deputy deans, heads of departments, professors, faculty members, student leaders, brother and sister in the hall, and our virtual audience joining us via IIUM ITD YouTube channel. We would also like to record our sincere appreciation to and welcome to our exhibitors, namely Pahang State Forestry Department, Kuantan Fire and Rescue Station, Unggul Medic Sendirian Merhad, Lab IBF, and National Anti-Drug Agency. Before we commence, let us begin with a dua to seek the blessing of Allah Subhanahu wa Ta'ala. For this, I would like to invite Assistant Professor Dr. Muhammad Azri bin Abdul Jalil from the Department of Basic Medical Sciences for Nursing, Kuliah of Nursing, and a member of PLANETIIUM to lead the recitation.
Al-Fatiha. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين حمدا يفيد بسم الله يا ربنا لك الحمد كما ينبغي لجلال وجهك كريم عظيم سلطانك اللهم صل وسلم على سيدنا محمد صلاة تنجينا بها من جميع الآفات وتقديرنا بها جميع الحاجات وتطهرنا بها من السيئات وترفعنا بها عندك على الدرجات وتبلغنا بها الأقصى الغايات من جميع الخيرات في الحياة وبعد الممات وعلى آله وصحبه أجمعين. اللهم اجعل جمعنا هذا جمعا مرحوما وتفرقنا من بعده تفرقا معصوما ولا تجعل اللهم فينا ولا معنا ولا من يتبعنا شقيا ولا مطرودا ولا محروما. اللهم اجعل خيرا أعمالنا آخره وخير أعمالنا خواتمه وخير أيامنا يوم نلقاك فيك. اللهم اغفر لنا ذنوبنا ولإخواننا الذين سبقونا بالإيمان ولا تجعل في قلوبنا غلا للذين آمنوا ربنا إنك رؤوف رحيم. اللهم إنا نسألك رضاك والجنة ونعوذ بك من سخطك والنار. اللهم عفو كريم تحب عفو عنا وعن والدينا وجميع المسلمين والمسلمات والمؤمنين والمؤمنات. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار. وصلى الله على سيدنا محمد وعلى آله وصحبه وسلم والحمد لله رب العالمين.
Amin, amin, ya Rabbil Alameen. Thank you, Dr. Muhammad Azri. Ladies and gentlemen, I'm Assistant Professor Dr. Muhammad Hanif bin Jailabdin, and it is an honor to serve as your MC for this distinguished occasion. On behalf of the committee, we sincerely appreciate your presence today, Thursday, 30th March 2025, 12th Ramadan 146 Hijriah, as we convene for the second Professorial Lecture of 2025 at IIUM Kuantan. This morning, we gather to celebrate the scholarly contribution of Professor Muhammad Lokman bin Mat Isa, a leading figure in planetary health and director of PLANETIIUM. His work continues to shape sustainable well-being initiative in Malaysia and beyond. Without further ado, I would now like to invite Assistant Professor Dr. Rizwanul Hakim bin Abdul Razak, Deputy Director of PLANETIIUM, to deliver his welcoming remarks. Please, welcome.
Honorable Professor Dr. Samsul bin Rahman, Member of the IIUM Board of Governors, Associate Professor Dr. Razman bin Muhammad Ros, Deputy Campus Director, Student Development and Student Support Services, our celebrated Professor, Professor Technologist Dr. Muhammad Lokman Mat Isa, Director Institute of the Planetary Survival for Sustainable Wellbeing, Assistant Professor Dr. Muhammad Shukri bin Babar, Acting Chairman for Professorial Lecture, Professor Dr. Suzana binti Abdul Rahman, Professor Kuliah of Allied Sciences, esteemed professors, deans and directors, respected academicians, researchers and industrial experts, ladies and gentlemen, Assalamualaikum warahmatullahi taala wabarakatuh, and very good morning. It is my great honor and privilege to welcome each and every one of you to this momentous occasion, the Professorial Lecture by our esteemed scholars, Professor Technologist Dr. Muhammad Lokman bin Mat Isa. On behalf of the Institute of Planetary Survival for Sustainable Wellbeing, PLANETIIUM, I extend my deepest gratitude to all of you for joining us today to celebrate the remarkable academic journey and invaluable contributions of Professor Muhammad Lokman in the field of reproductive technologies and planetary health. On a personal note, I believe that young minds at PLANETIIUM honestly regard Prof. Lokman as a mentor in shaping our research horizon and paradigm. It is a great moment for me, especially and personally, to speak today during my PhD supervisor's professorial talk, a testament to the lasting impact of his mentorship and guidance in my academic and research journey. The topic of today's lecture, from pluripotent stem cells to somatic cell nuclear transfer, advancing reproductive technology for sustainable planetary health, is both timely and essential. As we navigate the complex challenges of sustainability and also the environmental change and health, groundbreaking advancement in reproductive technologies hold immense potential in shaping a future where human and planetary well-being coexist harmoniously. Prof. Lokman's pioneering research stands as a beacon of innovation, guiding us to responsible and ethical scientific progress that aligns with planetary health, the interconnectedness between human and also the natural ecosystem that we depend on. At PLANETIIUM, our commitment to planetary health, sustainability, biodiversity, and scientific explorations aligns perfectly with the themes explored in today's lecture. We believe that collaborative effort between researchers, policymakers, and industry leaders are vital in translating this advancement into impactful real-world solutions. This commitment is deeply rooted in the Tawhidi epistemology, which emphasizes the oneness of Allah as the foundation of knowledge and the integration of science with divine guidance. The pursuit of knowledge, particularly in reproductive technologies and sustainability, should be driven by the higher purpose of serving humanity and preserving the balance of nature as commanded in the Quran. PLANETIIUM was launched in 2023 as a response to the growing need for transdisciplinary research that bridge environmental sustainability, health, and well-being. Our institute focuses on 4 key research areas: climate action, biodiversity and conservation, food security, and health empowerment. Through these core themes, we strive to generate knowledge and solutions that foster resilience and sustainability for both humanity and the planet. One of the PLANETIUM's most groundbreaking initiatives is the Frozen Zoo, first of its kind in the Southeast Asia. This effort is established as an agile response to the urgent need for genetic conservation amid increasing biodiversity loss. The Frozen Zoo serves as a genetic repository, cryopreserving viable cells, gametes, and embryos of endangered species. This initiative supports wildlife conservation, facilitates reproductive research, and enables species restoration and population recovery. By integrating biotechnology with conservation strategies, the Frozen Zoo plays a crucial role in protecting biodiversity and ensuring the survival of threatened species for future generations. Apart from that, PLANETIIUM is also home to several dedicated research centers. The first one is SUSTAIN, Sustainable Technology and Innovation for Food Security, that is responsible as the forefront to enhance the food security through sustainable practices and innovative solutions. Another one is BASICS Biosphere Courtyards, serve as a hub for ecological and community-based research focusing on environmental conservation and sustainable agricultural practice. Meanwhile, SINA, Special Needs Community Wellbeing Training and Research Center, is dedicated to supporting individuals with special needs through research-based interventions, training programs, and community empowerment. Recognizing the intricate relationship between humans and nature, we have one Nature's Balance Gallery that is responsible to promote biodiversity conservation and environmental awareness through research and education. We have also Ecosphere or Sphere Ecospheres Microbiome Center, explore microbiome science to improve environmental and human health. And the last one is Earthling, Planetary Health Education Learning Program that integrates planetary health learning program and outreach activities to inspire future generations to take part in sustainability effort. In this regard, scientific progress must always be aligned with ethics, morality, and sustainability, ensuring that our advancement contribute to the Maqasid Sharia. Prof. Lokman's work is a testament of how cutting-edge research can be harmonized with this principle, driving innovation while upholding our responsibility as Khalifa of this earth. His research in reproductive technologies not only enhances human health, but also contribute to biodiversity conservation, genetic preservation, and also ecological balance, which are the components of the planetary health. I would also like to take this opportunity to extend our heartfelt appreciation to all institutions, organizations, and individuals who have contributed to the success of this event. A special note of gratitude goes to the academic support services of the Office of the Campus Director and the dedicated committee from PLANETIIUM, whose hard work and commitment have made this event possible. Your presence today signifies the vision and unwavering commitment and dedication to the advancement of science for a better and sustainable world. As we conclude, let us take a moment to pray for our celebrated professor, Professor Technologist Dr. Muhammad Lokman bin Man Isa, and for all of us gathered here today. May Allah subhanahu wa ta'ala bless him with continued success, wisdom, and strength to further his contributions to knowledge and humanity. May this lecture inspire new discoveries, fruitful collaboration, and the pursuit of knowledge that benefits both people and the planet. May Allah grant us all sincerity in our undertakes, guide us to make impactful contribution to society, reward our effort in this world and the hereafter. Amin. Once again, massive congratulations to Professor Technologist Dr. Muhammad Lokman bin Isa on this well-deserved milestone. We look forward to an enlightening and inspiring lecture from him that will surely spark new discussions, ideas, and collaboration. With that, I warmly welcome you all, and may this session be fruitful and enriching for everyone. With that, Assalamu alaikum warahmatullahi ta'ala wabarakatuh.
Thank you, Dr. Rizwan. Before we proceed with the lecture, I'm pleased to invite Professor Dr. Susanna binti Abdul Rahman, Professor from Kuliah of Allied Health Sciences, to deliver the biography and introduction of our distinguished speaker. Please welcome Professor Susanna.
Thank you very much, Brother MC, Dr. Muhammad Hanif. Professor Dr. Samsul bin Draman, member of the IIUM Board of Governors. Associate Professor Dr. Razman bin Muhammad Rus, Deputy Campus Director, Student Development and Support Services. And our distinguished speaker, Professor Technologist Muhammad Lokman bin Man Isa, Director, Institute of Planetary Survival for Sustainable Well-Being, and Assistant Professor Dr. Rizwanul Hakim bin Abdul Razak, Deputy Director of PLANETIIUM, and Assistant Professor Dr. Muhammad Shukri bin Babar, our Acting Chairman for this Professorial Lecture. So we also extend our warmest greetings to all that are here, staff and students, brothers and sisters in this hall, and also those online. So I am Susanna from the Kuliah of Allied Health Sciences, and it is my pleasure today to be reading the biography of my good friend, Prof. Lokman, or Prof. Loki, as we all affectionately call him. So with your permission, Prof. Prof. Technologist Dr. Muhammad Lokman Mat Isa was born in Arau, Perlis to Mat Isa Mat Nur and Salmiah Ibrahim. His parents, collectively known as Salisa Ibn Nur, instilled in him strong values of education, discipline, and scientific curiosity from an early age. He began his early education at Sekolah Tengku Budria, where he excelled academically and served as a head prefect. He later continued his studies at Sekolah Menengah Kebangsaan Agama, Arau, a school known for its rigorous academic curriculum and emphasis on both scientific knowledge and Islamic values. As for his academic journey, after completing his secondary education, he pursued a bachelor's degree in biomedical science at University Kebangsaan Malaysia, where he gained a strong foundation in human physiology, pathology, pharmacology, microbiology, immunology, molecular biology, and genetics. Throughout his undergraduate studies, he developed a keen interest in cellular interactions, biochemical pathways, and disease mechanisms, with a particular focus on reproductive sciences and regenerative medicine. His research explored the molecular underpinnings of cellular processes, laying the groundwork for his future contributions to biomedical innovation and clinical applications. Following his undergraduate studies, he continued his master's degree at UKM, where he specialized in pharmacology and nutrition-related biochemistry. His master's research focused on the effect of ascorbic acid in the regeneration of alpha tocopherols in lipid emulsion for parenteral nutrition. This research was significant in understanding how vitamin C, the ascorbic acid, plays a role in maintaining the stability and efficacy of vitamin E, alpha tocopherol, in lipid-based intravenous nutrition, a crucial aspect of clinical nutrition and critical care medicine. With a strong foundation in biomedical sciences and pharmacology, Prof. Technologist Dr. Muhammad Lokman pursued his doctorate in philosophy at the University of Sheffield, United Kingdom. His doctoral research focused on human germ cell development and spermatogenesis from stem cells, an essential area in reproductive biology and regenerative medicine. His work explored the molecular and cellular mechanisms involved in the differentiation of germ cells from stem cells, providing new insights into male fertility treatments and reproductive health advancements. As for research and his academic contributions, with expertise in molecular and cellular technology, reproductive stem cells, and assisted reproductive technology, ART, his academic journey at the International Islamic University Malaysia has been marked by diverse research endeavors spanning human, animal, and plant biology. His research contributions extend across stem cell research, germ cell differentiation, infertility treatments, and regenerative medicine, and with a strong emphasis on developing innovative reproductive technologies. Currently, Prof. Technologist Dr. Muhammad Lokman's research focuses on planetary health, a transdisciplinary initiative that integrates biomedical sciences, environmental sustainability, and conservation efforts. In a groundbreaking achievement, he successfully established the Frozen Zoo, the first of its kind in Southeast Asia, fostering regional networks dedicated to wildlife conservation and genetic preservation. The Frozen Zoo at IIUM serves as a biorepository for cell lines and biological samples from endangered species, providing innovative solutions to mitigate biodiversity loss and extinction risk due to climate change. His expertise in conservation biology is further recognized through his role as a member of the Expert Working Group on Endangered Wildlife Species, EWG-EWS, underscoring his commitment to safeguarding genetic diversity, advancing species preservation strategies, and integrating cutting-edge biomedical technologies into conservation efforts. As for academic leadership and mentorship, in addition to all his research, Professor Technologist Dr. Muhammad Lokman is a dedicated educator, mentor, and academic leader. Since 2017, he has actively supervised postgraduate students, guiding them in biomedical research methodologies, critical thinking, and scientific innovation. He graduated his first PhD student in 2017, marking the beginning of his contributions to postgraduate training and mentorship. To date, he has successfully supervised 6 PhD students as the main supervisor and 6 PhD students as a co-supervisor. Additionally, he has mentored 4 master students as the main supervisor and 5 master students as a co-supervisor, contributing significantly to the advancement of research in biomedical sciences, reproductive biology, and regenerative medicine. Beyond supervision, Prof. Technologist Dr. Muhammad Lokman has been actively involved in securing research grants, playing an important role in funding and supporting cutting-edge studies. His ability to secure competitive funding has allowed for the expansion of research in stem cell technology, assisted reproductive technology, conservation biology, and planetary health. These grants have supported both fundamental and applied research, fostering scientific discoveries and real-world applications in health sciences and biodiversity conservation. His research productivity is reflected in his extensive publication record, with numerous articles published in high-impact, peer-reviewed journals. His scholarly contributions cover a wide range of topics, including stem cell research, germ cell differentiation, reproductive biotechnology, cancer biology, and planetary health. His publications are widely cited, demonstrating his influence in biomedical and environmental sciences. In addition to his publications, Prof. Technologist Dr. Muhammad Lokman has contributed to intellectual property development, securing several patents related to health sciences, researches, and innovations. His contribution extend beyond academia through public engagement, science communication, and policy discussions. He actively participates in international conferences, academic collaborations, and workshops advocating for evidence-based medical practices, sustainability in health sciences, and interdisciplinary research integration. His leadership and research contributions continue to shape biomedical sciences, conservation biology, and planetary health initiatives, making a profound impact on both academic and global scientific communities. So congratulations, Prof. Technologist Dr. Muhammad Lokman. We are all very proud of you, and we wish you all the best. Thank you, and Assalamu alaikum.
Thank you, Professor Susanna. Ladies and gentlemen, before we begin the lecture, let us watch a short montage. And now, the moment we have all been waiting for, it is my great honor that I invite Professor Technologies Muhammad Lokman bin Mat Isa to the stage to deliver his professorial lecture entitled From Pluripotent Stem Cell to Somatic Cell Nuclear Transfer, Advancing Reproductive Technologies for Sustainable Planetary Health. Please welcome Professor Lokman.
Oh, ni dia punya. Yang ni. Okay. Alright, Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahi rabbil alamin wa bihi nasta'in wa ala alihi wa sahbihi ajma'in. Allahumma salli ala Sayyidina Muhammad wa ala alihi wa sahbihi ajma'in. First of all, I would like to thank Professor Dr. Samsul bin Draman, Professor Dr. Razman, Associate Professor Dr. Ridwan, Assistant Professor Dr. Muhammad Shukri, and Professor Dr. Susanna for having time for me in these programs, and then join me with to celebrate this professorial lecture. Okay, so I am very honored to having standing here after a long, adventurous journey of my study and as a lecturer and as a researcher. So today I'm having the opportunity to share with you guys about the whatever that I have done about maybe more than 20 years in these academic worlds, and then perhaps it will give us some kind of new knowledge or added knowledge to every one of us. Okay. So I would like to start my talk with that title is From Pluripotent Stem Cell to Somatic Cell Nuclear Transfer, Advancing Reproductive Technology for Sustainable Planetary Health. Well, actually, when I was in doing my degree, my bachelor degree in biomedical science, there is a history, some kind of, I cannot say, is it like a good or bad, but it's happened already. It is like a sin that when I was in a final year project, I have to choose, you know, what kind of research that I'm going to do. But unfortunately, I might say good to it, this kind of word. Unfortunately, I've been chosen by the faculty some research related to ectoparasites of the Timur Laut. Okay, so I have to do those kind of research that are more in anatomy. So I feel like, oh no, it's not me, because I don't really keen on having this kind of research. So I went back to the supervisor, and then I said that I cannot do that research. And then she said that, no, you have to do, or else you have to find a replacer for that. And I'm a bit like panic, shocked. Who's going to replace me? You know? At the end of the day, I go to Deputy Dean Academics, Prof. Jamal, during that time. And then he is the one, he was doing research related to forensic and reproductive. So after several conversations with that lecturer, so at the end of the day, they agreed. And then Prof. Jamal asked me either to do research related to reproductive or forensic. So I chose reproductive, and from that moment, I love the reproductive science. So that's why I continue my research more on this field. Not just human, but now I'm doing more on animals, on endangered species. Okay, I think that's first of my introductions about my experience on how I jump into this field. Okay, when we're talking about reproductive, it is something that I guess that everyone aware about reproductive, and it's very important, and then everyone loves to have a family, isn't it? Okay, so when we're talking about reproductive technology, it is like the reproductive problem in ensuring the continuity of lineage and inheritance. But, and it is important, the problem happen in reproductive to both sexes, men and women, and also transgender as well, okay? And they have a lot of procedure. In assisting reproductive, we have basic to complex procedures, and it covers like essential treatment or even desire. You want to have bigger, you have to have whatever the things in reproductive you can do nowadays, okay? So that is why we as a Muslim in this world, we have to be guided by some ethical to hinder the misuse of this technology. As in Maqasid Sharia, number 3, we say that hifzul nasal, protections of lineage or progeny. So safeguarding family structure, lineage, and morality through laws on marriage, inheritance, and reproduction. I will share with you the technology that the world achieved today. There is a lot in reproductive. And then we can see how far the application and perhaps the complications toward that. Okay, first of all, I will tell a bit about what I've done in my PhD student, Lesson from the Past. So the title of my thesis previously is related to developments of post-meiotic germ cells of the sperms from human embryonic stem cell in vitro. Way back 15 years ago, during that time, the technology is about to start in the reproductive. So the stem cell is the main issue, the main player during that time. So there is a several group that doing research in stem cell research, and we happen to have to do research related to gametes, yeah, to a reproductive system. And we are working, we were like challenging each other, like during that time, Sheffield, Manchester, and then Newcastle are working, fighting each other to find the new latest findings related to gamete differentiation from the stem cell, a human embryonic stem cell. So as you can see that the gamete is the haploid cell. So the haploid cell is something that very crucial to see because of other group that using differentiation of stem cell are looking on somatic cell, which is diploid. So this is the challenge part that we were having during that time. Okay, as you can see here, this is like a very original about the stem cell, human pluripotent stem cell. So normally, Okay, normally what we get from the stem cell is, where we get the stem cell during the pregnancy is during the blastula, yeah, the blastula stage. So during that stage, we will take the cell, one cell from inner cell mass here, and then we grow in the media, in the specialized media, and then they will become embryonic stem cell. This embryonic stem cell capable to produce all types of the cell in our body. Okay, they're capable to produce all type of the cell, and then at the end of the day, they will produce the functional cell. Okay, the cell that we are having now is the functional cells. The stem cell is something like the cell that not yet been given the function. Okay. Previous thought, we said that when they enter one pathway, they cannot go to another pathway. If they are meant to become a brain, they cannot produce a blood. Previous understanding. But nowadays, there is a lot of findings related to stem cell differentiation, which is like they can bypass the past. You know, the pathway they can enter. They can enter to another laboraya, to another highway to become another things. Okay, let's say for example, if they enter the endodermal stem cell, soon they can become the skin cell. Okay, because ultimately they will produce the so we are asking back, what about the previously that we say there is no interchange? So this is what we call, we have to learn and relearn. Learn and relearn or unlearn. So there is like a sustainability word for this. Okay? And then they come with a new terms. Trans-differentiations. Okay? Previously there is no term on that. So now they have a new term, trans-differentiations. Bukan transgender. Okay? So this is how we see the evolvement of the knowledge itself. The evolvement of the knowledge is not just like, it will not the same 25 years pass, you know. So it will change. That's why we have to keep on learning, keep on getting the informations, and then like long life learning is the most important part. Okay? So we have got information about embryonic stem cell from the human or from the animal is able to produce any type of the cell of our body. So that's why we are using embryonic stem cell during that time to produce the gamete. Okay, this figure is what I've did myself. Okay, so if we can see that from here, we can see the germ cell cycle here. We can get the embryonic germ cell here from the fetus, okay, from the fetus. And embryonic stem cell itself is not the pluripotent stem cell. It is multipotent stem cell, which is they cannot produce any type, they cannot like embryonic stem cell. But for embryonic stem cell, they can produce more types of the cell, any types of the cell in the body. And then we also have the spermatogenic stem cell, which is they're going to produce the pathway to produce the sperm cell and related. Okay, if we can have a look here, from embryonic stem cell, this is like what we try to achieve in my research previously. We have embryonic stem cell, and then we have the primordial germ cell precursor, and also we have primordial germ cell, and we're going to have like germ cell at the end. I'm not expecting to get the sperms and ovum at the end of my study because it will take a long time. So my study was stopped at germ cell-like cell during that time. Okay, if you can see here, there is a fetal development and also neonatal, pubertal, and adult development of the sperm. What I can see here, when you do the lab test or when you do the lab identification using the stem cell, you have to bear in mind that you have to mimic whatever physiological happened. Okay, because during my time, they try to have like a transfect certain enzyme at a certain gene into the embryonic stem cell to produce the specific cell, but they are not following the physiological level, the physiological process. Let's say, for example, from the embryonic stem cell, they will have the primordial germ cell, and at the end of the day, they will have the sperm. But with the technology of the transfection gene, you can get the embryonic stem cell directly to become sperms. So they are not following the normal transformation. So there is a lot of ethical that been discussed about the thing during that time. And even now, we are still discussing about how good our approach on doing that kind of research. So we can see here, the best sign is to follow the natural development, the formation of male gamete in the testes. So we have to follow whatever that happens. So that's why during that time we think about what the natural things. So the developments of the sperm itself in the seminiferous tubes. So that's why we try to choose, we try to mimic them, and then we try to use the seminiferous tubule from the neonatal mouse testis. So this is like a marker. For the molecular, the marker is most important thing because we want to see whatever the specific markers that represent the stage of the transformation or differentiation of the sperms. So we have to identify, and then we have to check on the sample that we grow, either those kind of sample being appeared. So there is a technology that we link with the green fluorescent protein so that they will appear green or red if we tag with the primers on that. Okay. So to be honest, during this time, this marker is most important and it's most popular, but after 25 years, there is more markers that more significant or that more relevant to this level of the differentiation. Okay, I was doing my research for using the human cell. So this is like a remarkable result that we found, that we can get the haploid cell. We can get the haploid cell that color greens, so that because of that, we can say that our media that being introduced to this sample can manage to produce the cell, can manage to differentiate the cell to become haploid cell. As you know, the haploid cell is the 23 chromosome number instead of 46 chromosome numbers. So, but to be honest, during my time, I was getting about very little percentage of the cell that producing or differentiated into the haploid cell. So my supervisor said, okay, that's enough. You can graduate now, because because I can get this result. Okay, so and one more thing, when we're talking about the gamete cell, we are talking about the imprinted genes. Imprinted gene is something that the genes, the expression that just working on one, okay, on one gene. There is no not the haploid, not the diploid genes. Okay, only for maternal or paternal only. So that is why during the lineage of the inheritance, sometimes you can get kind of characters that just coming from your mother or your father. This is because of if imprinted gene of the paternal means that the gene from the father is being imprinted, and then they will not being expressed, so that the mother, even though it is recessive, it's able to produce the character or the phenotypes of the character. So when we're talking about the haploid cell, the most important part is we're talking about the imprinted gene. So during that time, I list down several of the imprinted gene, and then I try to identify how the pattern of expressions of this imprinted gene during the differentiation process of the human embryonic stem cell to produce the gametes. Okay, at the end of my research, I prolonged or I expanded the culture of the media with the specific media from the neonatal mouse testis condition media and plus the hormone. So with the prolong of 45 days, I managed to get the result that I showed just now about the fish result for the haploid cell. Okay, this is the neonatal mouse testis tube and seminiferous tube, and then I insert the cell, the single cell here, and then we hope that the cell can grow inside the seminiferous tube here. But unfortunately, the next day, the cell dispersed and become 2Ds, yeah? And then when we discuss a bit, and then the idea of getting secretum, or the idea of getting the media culture that being produced by this neonatal mouse testis, we just put it in to become conditioned media. So this is how we think and critically think on how to use this method. So Alhamdulillah, we managed to get the good result during the time, and then I try to compare whatever I've done to the natural or the physiological happen in the body with the one that we do. But during that time, we are not doing any transfections of the gene or gene manipulation, because in our school of thought, we try to avoid any disturbance of the sequence of the DNA, because we don't want to produce the transgenic human or something during that time. Okay, we are like modifying the chemical in the media culture to get the differentiation happen. Okay, so that's all about what I've done, what I've gained about the knowledge related to my research. So now it's about like the latest finding that we are having in this world about the differentiation process from 2010 until now. So if you have a look here, early research and proof of concept. In early 2010, pluripotent cell could differentiate into germ cell. 2009 study discussed the potential of pluripotent stem cells to form egg and sperm, and scientific possibilities and ethical consideration. So starting on 2010, we already starting to talk about the ethical issue related of the ability of the stem cell to produce the gametes. Yeah? Why ethical issue is so important in this kind of research? This is because inheritance, the lineage. Yeah, if you differentiate the stem cell to a cardiac cell, it's just for your own self. It's just for one human. But if you're producing the stem cell to sperm and egg, more complication, more incident, and more problem perhaps, or even more advantage, to be honest, that we can gain from this research. So that is how Maqasid Syariah, Tawhidi epistemology, all those kind of elements should be in our mind when we are doing this kind of research. Yeah, because of in Tawhidi epistemology itself, there are possibility of us, because to be honest, after like we are now in postmodernization era, you know, because of previously the modernization era is being, is finished already, and then it is just like we can say it is like a fail, because of there is a lot of problem that we have been facing here, and then we now in the post-modernization we try to introduce a better understanding of science, okay, like quantum physics, and in biology, even in my field, there is a lot of things that we have to learn, relearn, and unlearn or something, you know. So so that is why when we have the Tawhidic epistemology, it is very important to produce the Islamic way of scientific research. Okay, we can have like a kind of option to guide us to produce a better result of our research. Okay, so if you can see here, advancement in vitro gametogenesis. So Japanese scientists generated functional male and female mouse germ cell from iPS cell. Okay, after maybe about 2 or 3 years, I've gone from my PhD university. So they are producing induced pluripotent stem cell by what do you call it? Overexpression of 4 types of genes. So that kind of gene producing the embryonic, pluripotent stem cell. That similar functions of the embryonic stem cell. And then, when they have the embryonic stem cell, they produce the oocyte, functional oocyte, and then producing the healthy offsprings, so that we can have the oocyte from the induced pluripotent stem cell in the mid of 2010. Recent development and ethical consideration, they keep on refining the technique, because the most important part is to make a standardized. Like medical doctor, clinician, they will go for standardization. We reported a lot. We can get this and that, but there is no standardization. One institute can get it, but some institute cannot get it. The reproducibility is not there. So that is how, even though in journal they say they manage to get this and that, but sometimes there is a lot of question that we can ask. Okay. We also have the regularity inside. HFEA reported that technologies for in vitro gamete IVG are nearly viability. Okay, so means that it's about like in 2025 we are about to have the in vitro gametes technology. So this is a new revolutionized fertility treatment. Recommendation for regulator oversight, safe and ethical application. So when we're talking about the in vitro gametes, it's something related to, we are producing, previously we have IVF, in vitro fertilization. So we have gamete and sperm, we have sperm and ovum in our hand, and then we fertilized it outside the body. But now we don't have sperm, or we don't have ovum. So that's in vitro gamete. We try to produce the sperm or ovum, the gametes, from the skin cell, from the somatic cell. So this is the latest technology that we are having now. Okay? We can do a lot of things, like we remove the nucleus of the the oocyte, and then we reprogram the sperm and stuff, and then we can get the gamete that we want. Okay, that is ahead of the IVF. It is IVG, in vitro gamete. There is a lot of implication that we can discuss a bit now. Okay, so this is same-sex reproduction research done by researchers in China. Were creating mice from 2 male parents using gene editing techniques and infertile offspring. So nanti the group of gay can have, because previously they have a group of homosexual that having their own son, their own offspring, but by having the oocyte donor, you know, they still have a oocyte donor. So it just, what do you call it, contribution for one part only, from one couple only, paternal or maternal. I don't know paternal or maternal which side, okay? But then nowadays with the technology, both can donor their sperms, and those two sperm can fertilize, can be fertilized inside the ovum that being remove the nucleus and become the gamete. It's a bit scary, isn't it? Okay, so that is why the Chinese researchers have done this kind of research, and of course there is a lot of ethical hurdles that we have to think about. You know, what kind of the family constitution at the end of the day. So as I mentioned just now, we have ongoing challenge and future direction related to the reproductive technology. We have like a mouse model versus human model. Sometimes we say yes to mouse, but maybe when we want to do in human clinical trial and stuff, maybe it's not working to the sample or to the gamete of the human based on physiological and anatomical biochemical differences perhaps. So ethical and social implication, the potential for new family structure. Okay, the potential for new family structure. So if a gay can contribute both sperm, so what's going to be next? Okay, the implication of genetic parenthood without traditional reproduction. So now kita tak payah kahwin dah. Okay, we can just donate our gametes and then we can get like express. And even now they are talking about producing the synthetic or artificial ovary, artificial uterus, yeah? So that even we can have our baby, like we put an egg on the oven, incubator, egg of the chicken in the incubator, at the end of the day we can get the baby of chicken inside the incubator. So macam cerita yang dah ada tu is going to happen after this. Okay, robust ethical guideline to navigate this emerging possibility. So that's why from time to time there is like an inter-religion talk about this. How far we going to, you know, how far we can proceed this kind of research, because at the end of the day maybe this kind of research perhaps can able to change the whole institution of the human beings, you know. This is for human actually, but for animal there are other things. Okay, for human we have to really focus. We have to really think about this meticulously because of the effect is very vast. But for animal, maybe we have to work on. We have to continue because of there is a lot of endangered problems, you know. We have to, in terms of the, to ensure that they are keep on living, because the numbers sometimes, some of the animal is decreasing. So, in summary, we say, from 2010 to the present, stem cell research has made remarkable progress toward producing gamete cell. While this development holds promise for addressing infertility and expanding reproductive options, they are accompanied by complex ethical, technical, and regulatory challenges that require careful consideration as the science advances. Okay? So this is like one whole thing about 2010 until 2025 of research related to embryonic stem cell producing the gametes. Yeah? But to bear in mind here, most of this research have been done using animals. Yeah? Okay? So, if you're having here, key techniques used in gamete derivation in this time, 2010 to 2025, we have induced pluripotent stem cell. Adult somatic cells are reprogrammed to an embryonic-like state, enabling their differentiation into germ cell. Embryonic stem cell derived from early stage embryo. This cell can differentiate into primary germ cells and become sperm and egg. In vitro gametogenesis, IVG, the process of generating functional gamete outside the body by differentiating IPS and ESC into mature sperm and egg. And then we have these tools, yeah, CRISPR-Cas9 gene editing. This is a tool to ensure the possibility of getting gene that being transferred, the possibility is higher. Because previously, if we don't have this CRISPR, the possibility of getting the right attachment of the primer that we have is very low. So sometimes the failure is high. Okay? So major genetic factor in gametogenesis, we have BLIM-P1, PRDM14. Okay, like previously we have like, during my time is PRDM10, but now it's 14. So they have identified another more about the markers of the gametogenesis. Okay. Okay. Recent controversies and ethical concern in terms of ethical and social implication. We have designer baby, like we can program. If orang Melayu want to have their baby with a green eye, with a high intelligence, with a tall and dark skin, tall, dark and handsome, something like that, you can program your baby by this technology. Okay? And another thing is same-sex reproductions. Yeah, like what I mentioned just now, from the IVG technology, both can contribute to produce the offspring. So think about that. And then embryo farming concern, maybe kita buat macam, there is a problem like ikan beta, ikan beta, they are farming. There are farms that produce the ikan beta in a huge number and then they can sell it. Last time, during my time, ikan beta we can get all in the paddy field. But nowadays, when they become famous, viral, so the price is very high. Same goes to the embryo farming concern after this. Maybe the human, they can just produce the embryo and then they can just sell it in the black market and everything. So what about the human constitutions? Because we are Khalifa, we are the vicegerents in this world, but then we are about like, can be sold in the markets, just like that, okay? Back to like the era of the Jahiliyyah, that we can, like the human being can be sold as easy as that, okay? Maybe it is like the cycle will go back, turn around, okay? And then we have the regulatory challenge, legal uncertainty. Existing reproductive law may not adequately address IVG. Countries like the UK are leading in defining legal frameworks. UK again, but we produce our legal also in Malaysia. Maybe IIUM can lead with that, with the elements of Tauhidic epistemology, inshallah. Okay, consent issue, involvement of donor cell for gamete creation, raise question about informed consent and genetic ownership. And then, of course, like I say, maybe we can introduce, we can make it popular about the Tauhidic epistemology. Okay, we can see here, the clinical trial in the next 5 years anticipated human trial for lab-grown gametes, IVG, under strict ethical guidelines, particularly in UK and Japan. Means that they are towards that in another 5 years. Fertility treatment, potential use for individual with infertility due to the chemotherapy, aging, and genetic factors. This is the good the goodwill, actually. We start with the goodwill, and after that, we manipulate it. Okay, that's what happened. For the first thing, we want to treat infertility for the people that don't have baby, blah blah blah. But at the end of the day, when we got some idea about that, and then some findings, some breakthrough, at the end of the day, we have another idea, another silly idea, not silly, another like nasty things. Oh, we can do things, we can do this. And at the end of the day, they have the idea, because they are working on having the rights of homosexuality. So at the end of the day, they say, see, we can produce the baby as well. So what the difference with us as a homosexual compared to the heterosexual? So at the end of the day, the community have to accept them. Okay? So this is the hard complication, the worst complication that we have to think about. For long term, personalized reproductive medicine, okay, as we are talking about the personalized medicine, so we can do the personalized reproductive medicine. Human genome editing integration, okay, now we can have like perhaps a new species, a new genetic differences of the let's say like design baby that we produce. So what can we say? Maybe a new human, you know? So bioethical framework, or this is like have to put in like SDG or something so that all the world can work together with this. So this is one case study. So to derive human primordial germ cell, like cell, from embryonic stem cell. For stem cell activity, for stem cell study, we like to use the word like cell, because we are not producing the real cell actually, just like cell. Okay? Methodology, scientists use human pluripotent stem cell and expose them to the cocktail of growth factors. They induce differentiation into that HPG-LC in vitro. Gene expression was tracked to ensure the cell mimic natural human germ cell development. So they're producing successfully generated human germ cell precursor from iPS cell, but in incomplete maturation, and then ethical concern arose due to the potential to create human embryo in vitro. So the impact, this was a milestone in human germ research, but highlighted technical and ethical issue. Open discussion about clinical application for infertility treatment. The second one, this one is the one that I mentioned just now, that being doing by the Chinese researcher in China to create mouse embryo using genetic material from 2 male parents. Researcher edit genomic imprinting region, which normally distinguish maternal or paternal. They combine the genetic material of 2 male mice to create embryo, implanted the embryo into female surrogate. So they can get the survival of the offspring, proving the same-sex production in theoretically is possible. But then many offspring had developed mental abnormalities and died early, maybe because of the issue with the DNA combination, ethical concern about applying this to the human reproduction remain unresolved. The impact demonstrates that it may be possible for same-sex couples to have genetically related children, raise serious ethical and safety concern about future human application. So this is about the public opinion on stem cell research that been doing the survey. Supportive view, what they said, infertility treatment offer hope to individuals with genetic infertility, cancer survivor and aging population. This is a good views. Same-sex parenthood provide biological parenthood option for LGBTQ plus couples. Okay, so there is a lot of people that support this view. Genetic disease prevention, potential to screen and edit out heritable disease during gamete production. So this is like a specialized medication. The one that oppose is the ethical dilemma, fear of creating embryos solely for research or reproductive use, design baby and identity concern. Okay. Okay. Gambar tu can. Okay, if we compare the embryonic stem cell, iPS cell, and IVG, embryonic gametes, so for the source for embryonic stem cell, we have from blastocyst, this one for the skin cell, and this one is derived from both of these. Okay, pluripotency means that the ability to produce in any type of cell, yes, and both are yes. Okay. Ethical concern, destructions of embryo, genetic reprogramming and safety issues, potential misuse in human reproduction, the gay issue here. Clinical potential, limited, this is high, and then this is the future. Gamete creation, possible in mice, in human is questionable, possible in mice, experimental in human, future goal for human fertility. Notable achievement, 2016, mouse gamete for ESC, 2016, mouse offspring for iPSC, 2024, the latest one, mouse embryo from 2 males. So here I list down a bit of the pathway of the human germ cell developments. They are working on this pathway. When we're talking about the pathway, sometimes like the researcher in molecular, they want to know what kind of thing that responsible, what kind of element, what kind of chemical, biochemical molecular that work on this. But sometimes somehow it works synergistically, you know, like honey. Okay, we cannot like extract and get the bioactive, and then we want to have the bioactive itself. But then the best thing is we consume as it is. Okay, that's the dilemma in the molecular research, because we want to see what the one thing that works. But sometimes when we get the one thing, tomorrow we test, there is a one thing prime. So they have another things. Sometimes when you want to play with the God, God also play with you. Okay, that's the issue. So this is like a several pathway that being studying on that. Okay, and again here, SOX9 is the most important pathway here. Okay, if you can see here, I just want to show you how the pathway is working well, and then this is to show the undifferentiated capability of the embryonic stem cell. So they're able to produce, to differentiate, to form different types of the cell, but they also have the capability to maintain themselves as undifferentiated. Maksudnya, they don't have they not go into any type of cell, they just remain as it is. So this is important because of it's like a backup. If something happens, they can differentiate into the cell that we need. Okay, So this is how good, how creative our God producing this kind of mechanistic, to ensure that it is well and safe, sustainable. Okay, so we have to do kind of work like this. I mean, we have to ensure that we also can produce those kind of things. So same goes to Nanook and SOX 2. They are also able to have like to ensure that those cell is in the phase of undifferentiated. So kalau if we have a look here for GDF9, for GDF, this is the pathway for producing spermatogenesis cell, yeah, the cell. So I just want to show you, this is like the one that we study with all these kind of elements that we want to ensure that the production is normal, yeah. So when we're talking about the imprinting gene, the expression in parent-specific manner, only the copy from one parent, either maternal or paternal, is active while the other copy is silenced. Controlled by epigenetic mechanisms. So epigenetic mechanism here means that they are not changing in terms of the DNA, but after the DNA. So that like when we want to produce the protein, we will undergo the epigenetics. And then there is two or more activity, which is DNA methylation and histone modification that happen to produce the imprinted gene. So that if the DNA being removes the OH group there, and then they will produce, the gene will not be expressed for that stage. Maybe on the next inheritance, they can produce it back. So that's what happened to the imprinted genes, yeah? Do not change the DNA sequence, but affect gene activity. So parent of origin-specific expression, one allele from either mother and father is active while the other is silent. So epigenetic regulation controlled by DNA methylation and histone modification at imprinting controls region center. Tissue-specific expression, some imprinted genes are actively only in specific tissue, particularly in the placenta and brain. Okay, development important, play critical roles in fetal growth, placenta development, and metabolism. Okay, here is like the center of the imprinted that work to methylate this kind of genes. Okay, let's say for imprinted parent, moms here, maternal imprinting, so the father will express the phenotypes. But if the father is imprinted, so the mother will have the phenotype. So that we can see that if the genotype is the recessive, so the recessive part can have the possibility to being expressed rather than the dominant one. So this is an example of imprinting genes. And then why it is important. Embryonic developments, imprinted genes regulate fetal growth, placental functions, and neurodevelopments. And then genomic balance, imprinted ensure a balance between maternal and paternal gene contribution. Disease association, disruption in imprinting can cause disease like cancer, developmental disorder, and metabolic syndrome. Okay, now again we have embryonic stem cell, but nowadays we can produce haploid embryonic stem cell. What is that? Embryonic stem cell that contain a single set of chromosome, unlike regular diploid ones. This cell produce a unique model for studying genetics developments. So they have 23 instead of 46 of the chromosomes. They still can have pluripotency capability. And then genetic simplicity, since they have only one copy of each gene, mutation and genetic changes are easier to study. Artificial derivation, typically derived from unfertilized oocyte, or we call it parthenogenesis, or create through genome editing. So this is how I want to show you after this, okay? So we can do we can apply this to the genetic research, drug screening, disease modeling, reproductive biology, and gene editing. Everything we can do now. Okay, how haploid ESG produce? Parthenogenesis, they will stimulating the unfertilized egg to divide and producing haploid. So means that the egg, if it's not being fertilized, they can be stimulated as well to produce embryo. Okay. Genetic manipulation, engineered diploid ES cell to become haploid through specific technique. Derivation from germ cell. So from the germ cell, they will get the haploid germ cell here. Okay. Challenges, genomic instability. Normally, haploid cell can easily become diploid. Limited viability, harder to maintain in culture compared to diploid. Ethical consideration, similar to the thing that we discussed previously. Okay. If you want to have like this kind of cell, so we can activate the blastocyst here. So from here we can get like two populations of the cell, which is one diploid and one haploid. And from this one, we remove the nucleus of the oocyte, and then we produce, we insert the sperm, and then the oocyte will become embryo. Okay, and then now we can have like a haploid embryonic stem cell, that from the paternal. And the other one is remove female pronucleus here, and then when we remove here, so it's only one, then from this one they can produce the embryo, and from embryo we can take the cell as well. So we have androgenic haploid embryonic stem cell. They are derived from the paternal sperms. The cell carry a single chromosome, not like paternal ones. Okay, they originate from the male genomes. One set of the chromosomes and pluripotents, and then reflect to the paternal specific imprinting patterns, allowing researchers to study paternal effect. How to do that? Spermatic is isolation, so they will get the spermatic, which is the round cell before producing spermatozoa, collecting immature sperm cells, the spermatid or spermatozoa, artificial activation, stimulating this cell to divide in nucleate egg, and then forming a haploid blastocyst. Then we can isolate from the embryo stage to get the haploid ESG. Let's say, for example, here, so we knock down certain of imprinted region here, so that's when we put this is like Chinese, what the Chinese do. They have the androgenic haploid cell here, and then they mix with the sperm here, and enter inside the oocyte here that no have nucleus anymore. So they will produce a they will produce the offspring of the two DNA of the male. Yeah. So this is how they do to get the baby of the homosexual. Okay. So application here, genetic research, reproductive biology, disease models, and all those kind of things. Challenge is similar, instability. And then we have parthenogenetic haploid, this one from the moms, yeah? The lesbian also can work, can do that as well. Okay? Haploid cell derived from the unfertilized egg through a process called parthenogenesis. This cell contains a single set of chromosomes from the maternal genome, ability to differentiate into various cell types. So derived exclusively from mother genetic materials, contain 23, can differentiate and display maternal specific genes. So first they collect the unfertilized egg, and then chemically or electrically stimulate the egg, and the egg will divide without fertilization. And then when they produce the blastocyst, they take the cell there, and then they will produce they grow there to produce the haploid embryonic stem cell. Okay, so the mother, the offspring with two DNA from two mother, so they will do similar things. So when they introduce first of all, they will activate the ovum itself, and then ovum will produce, will become embryo with haploid 1, okay? And then from that, they can take it out and then let them pregnant. So from that, they will have the haploid of the imprinted, of the parthenogenesis ESG. So it's similar, the field of application. Okay, one last part related to this is the human chimeras. Human chimeras is happen when during the pregnancy there is two ovums being fertilized, and then when the two ovums being fertilized, they're supposed to become the twins, yeah, the non-identical twins. But at the end of the day, once of the embryo being fused, you know, the other one, the dominant one, like eating the second embryo. So the consequence of that, the baby, the offspring, will have two sets of DNA. Okay? Because of that, when they have a two set of DNA, sometimes when they check the DNA, the genomic DNA of the skin, perhaps different compared to the genomic DNA of the lung, for example. Okay, but when it happened to the sperms or ovums, so they have several numbers as two set of the sperm DNA DNA genomic. Okay, so because of that, it will have a problem with the offspring. Sometimes when the father check their what they call it, their DNA genomic DNA, it's not similar to the kid because of maybe it's belong to the embryo that they engulfed previously. So they say that their wife is the one that unfilth. Okay, so because of that, maybe there is a divorce happen, something. Okay, that's why sometimes somehow in Islam, genetic identification is not so valid, because of that is kind of thing that happened. This is one of the proof that we have, like human chimeras, because we cannot check, we cannot easily identify, to be honest. Okay? So that's why in Islam sometimes sumpah is very important, yeah, not the genetic identification. Okay. Okay, let's say for example here. Oh, sorry. It can like, it is a normal one. So when the polar body here, they're being fertilized in polar body. So at the end of the day, the M2 and P2 will become ones here, yeah. And then like for example like this one also, P1 and P2 here, with the M1 and M2, and then they will have like two sets of the DNA genome here. So if you can see here, this boy having like a different color of the eyes. Okay. So two embryo merge into one during early pregnancy, and then cell from the fetus cross the placenta and persist in the mother. So it could happen in the bone marrow transplants as well. So the characteristic, genetic differences, different tissue may carry distinct DNA profile, may cause patchy skin to tones or heterochromia, different color eyes, but many cameras show no outward signs. Health effect often harmless but can lead to the autoimmune issue related to they have two types of the genes. Yeah, fertility impact, different gonadal tissue could lead to the reproductive complexity. This is the application of the camera research, creating human-animal camera to grow organ of transplant. So we do like transclone from human to animals. Again, this is ethical issue as well. Genetic study, diversity and developmental biology, disease modeling, animal models, and personalized treatment. What Islam say about this science? We have to gather to think about this. Okay? So this is the case study of Taylor Moore, the American singer. Oh, 10 minit lagi. Okay. So, right, we move again to the application of IVF in conservation of endangered species. Genetic diversity preservations. This is how I turn to human, to the animal research. Yeah, from the human, there's a lot of ethical issues. So I try to find a better way. So overcoming reproductive challenge with low fertility rate, do not breed well in captivity. Assisted reproductive technology can be combined with sperm, egg, and cryopreservation and stuff. So this is possibility that we can do in the IVF in conservations. So recent advance, we have the ocelot conservation effort, we have northern white rhinos, we have cheetah reproduction, and the challenge is the species-specific reproduction knowledge. Understanding of the reproductive physiology of each species vary widely, not always well documented. This is the thing that we are doing now, because of if you want to do the method to the monkey, maybe cannot similar to the Sumatran rhinos. Okay, and then it's not well documented. Ethical and ecological implication. Okay. Okay. Like I mentioned about previously, we have the chemical induction or genetic manipulation. Which one that you want to choose? So we have the iPS cells, and then we have the chemical reprogramming. But now, our group trying to do the SCNT reprogramming activity for our animal sample. When we're talking about the chemical, we're talking about the secretome in stem cell research. The stem cell secretome refer to the collection of bioactive molecules secreted by stem cell, including protein, lipid, extracellular vesicle, and exosome. Crucial role in cell communication, regenerative, and healing. So the component is just this one, okay, and this is the functions of this secretome. So they have cell-to-cell communication, modulating cellular behavior. So the main important of the secretome, we don't have to inject the cell. Yeah, we just injecting the liquid or the whatever the micro molecule that working on having the interaction. So we don't have to modify our DNA sequence and stuff, but we are doing a biochemical interaction. So we have secretome-based therapies, exosome engineering, and biomarker discovery. Okay, this is, for example, MSC, mesenchymal stem cell isolation from the tooth, and then expansion, modification genetics, condition media preparation. So we take the media, the one that being used by the cell, and then we inject to the human. Okay, this is like the secretome technology. Okay, then I enter to the frozen zoo, the one that we having now. Frozen zoo center that is equipped with the equipment is established in Malaysia, and it's the first in Southeast Asia. Related to the link to the activity that I have done previously, so I try to derive the cell line from the tissue is one of the unique aspects of the center, not at the other animal biobanking system. The method for cell derivation will optimize for every species of the cell. If you can see here, we can get the cell from the Sumatran rhino and also from the Malayan tiger. So this is like our collection. It keep on increasing the species, and then we have done. We are towards having this IPS cells and also the SCNT, the two elements, because we have somatic cell here. Okay. So this is the Sumatran rhinos, that are being extinct in Malaysia. So we have the cell lines of these Sumatran rhinos, and we are working on having somatic nuclear transfer technology to clone the cell to produce the gamete. Okay, we produce some papers and then cell line derivations, and then we are doing karyotyping of different cells as well. This is the challenging part because of lack of reference, and then the number of chromosomes is different. This one is 84, but the human is 46, kan? So we have to do the karyotyping lineage here. Is very very hard, to be honest. Okay. Okay, this is the cell of the three last Sumatran rhinos that we have now: Puntung, Kertam, and Iman. Kertam is the boy, the male, and the female is Puntung and Iman. And collection of the gametes, we have macaque, civet, gibbon, ovary, orangutan. Okay, and this is how we provide the cell line from tissue, primary cell, and then we produce the cell line and we cryopreserve. So this is the differences between primary culture, enzymatic, between the primary cell and also the cell line. Okay. Nowadays we are focusing on Malayan tiger because Sumatran tiger is totally extinct. Malayan tiger we have several, yeah, in the hundreds only the numbers. Okay. We try to do the IVF for Malayan tigers recently. Okay, this is the to show that we managed to get the two cell that producing the embryo from SCNT, which we inject the sperm. We inject the cell that we derive into the nucleus, into the oocyte that being removed the nucleus, enucleated. So we get the two cell that produced from the injection. Okay, our research project is collecting, and then we try to do the cloning, and then we try to produce back the animal, bring back the extinct animals. Okay, there is two approach. We have the, this one is the SCNT, and this one is the iPSC, yeah? Induced pluripotent stem cell. And then when we have the induced pluripotent stem cell, which is pluripotent, and then we can produce back the sperm and egg, and we fertilize it back. But this one is very straightforward. We use the cell that we have from the cell line, and then we remove the nucleus from the oocyte, and then we fertilize them, we inject the cell inside the oocyte, and they'll produce the embryo to get the baby. Of course, we need to have the surrogate mom for the badak sumbu that we have the cell. Okay. So when we want to do the SCNT, there is a several step that we have to think about, that we have to strictly understand, like activating the transformation of embryo and stuff. This is the thing that we are doing now in our lab. And then if we want to have like a sperm, ovum as well, we have to do the in vitro maturation of the animals, because of like in human, nowadays for IVF we measured, or we do the hormone stimulation to produce the matured oocyte, yeah, multi numbers. But then in animals we can do, because of it will cause stress for the animal because of a lot of injection that's supposed to be done. So what we do, we have to collect them, and then we do the maturation outside. Okay, this also having a lot of procedure, and also the media, types of media that we need to test to ensure that different species will have optimized method of maturation. Okay, and of course, genomic database of wildlife, there is not complete, less important, less being studied, also important to the farm animal. So this kind of thing also, we work on this, because tropic is the area that have most numbers of biodiversity. So that's why we take this opportunity to champion, to get the best as we can, the numbers of the genomic of the different species in our regions. Okay, this is San Diego Zoo. Maybe we have that IUM Frozen Zoo soon. That popular. This is the first one in the world, yeah. And they have like, see, you can see plenty of the storage facility that keep all the species. Okay, I wish I could visit one day in the San Diego Zoo. Okay, when we're talking about the planetary health, okay, tadi dari frozen zoo, from the animal, from human to animal, and then now to frozen zoo, producing the frozen zoo, and then we are looking to the interaction between human environments and the biodiversity. So we have the frozen zoo that brings me to the planetary health. Planetary health is interconnected of these three elements. Previously we were talking about public health, now we're talking about global health because of the urbanization, deforestation, climate change and stuff. And then at the end of the day, we are now talking about the planetary health with the interrelate the three elements of the biodiversity, human and environments. We try to find a solution, because the thing is happened, we cannot change it. But what we want to do, what thing that we can do to ensure that our planet still survive. When our planet survive, we will get the sustainable well-being. Sebab there is, some people say that we have to being well first to keep the planet. But our approach is we ensure the planet is survive, because we are the Khalifah, vicegerents that control the world. We ensure the planet is survive, then with the survival of the planet, we can live happily ever after. Okay? So that's why we have elements of 4 elements that we have, yeah? So holistic quintuple helix solution here with the industry, with the university, community, governments as well, to ensure that the planetary, or the balance between human, animals and environment ecosystem. Okay, this is the interconnectivity, biodiversity, health and climate change or environment. Biodiversity support human health and societal needs, including food security, energy development and development of medicine, pharmaceutical and clean water. Protecting biodiversity and climate change, protecting biodiversity can help us adapt to climate change. Healthy ecosystem will be more resilient and able to maintain the supply of ecosystem services on which our prosperity and well-being depends. And from health and climate change, mitigation and adaptation strategy will positively affect both climate change and environmental, thereby creating a positive effect on human health. Okay, this is like an example of the challenge when the human, it starts with the human and it's come back to the human again, yeah? human eating, consume endangered species, and then this endangered species a reservoir for this kind of virus, coronavirus, bats and stuff. So they will transmit back to you, to the animals, and then animal can transmit to the human when you consume it. Zoonotic infection here, and then human to human because the pandemic, and then cause the global health issue. Same goes to when you do the vegetation, encroach the lands, deforestation, climate change, and then bats will increase, those kind of things. It's like a circle. It starts with us. When we ask who to be blamed, it's the human to be blamed, to be honest, because we have akal that's different with animal. Animal only have brain. Okay, the akal that will lead us on how to ensure the planet is maintained, the planet is survived. So biodiversity and climate change, if the planet is okay, we can produce the good health, good well-being to the human. Okay, after that we have the PLANETIUM, the bonds of Institute of Planetary Survival for Sustainable Well-being on 2022. Okay, by safeguarding our planet survival, we secure a future and sustainable well-being for generations to come. Healthy planet, healthy you. Okay. So our vision is to be a distinguished research center of excellence for conducting multidisciplinary and transdisciplinary research and innovations involving health, biodiversity, and climate change for commercial and community empowerment. And our mission is to integrate the multi-transdisciplinary research involving health, environment, biodiversity, and climate change to produce relevant solutions for commercialization and community empowerment to ensure talent development is sustainable well-being. So this is our mother, our advisor for the PLANETIUM until now. It's better to start now than not to start at all. She is now the chair for National Planetary Health Action Plan Malaysia. So we have 4 research pillars in PLANETIUMs to enhance food security, climate action to overcome climate crisis, to improve health empowerment, and to conserve and sustain the biodiversity, to increase quality of life and sustainable planet and societal well-being. So we have 4 action, 4 real action. We have rescue, restore, and resurrect, and also reintegrate to ensure that we're going to find a solution for the planetary health issue challenge. Yeah. So we have quintuple helix that work together, which is industry, university. This is industry, integrated knowledge researcher for university, government, community to ensure the planetary health, balance between human, animal, environmental ecosystem. Frugal innovation, boleh remove gambar ni tak? Finding solution to planetary health challenge require frugal innovation, the use of low-cost technologies, and a shared commitment across all levels of community to protect both humanity and the earth for future generation. That's why in PLANETIUM we try to find, ataupun we try to produce the technology that can be used by all different types of community. Okay, we try to use the local technology, the local things that are available around us rather than we import the things from outside. That will cost a lot higher cost. Yeah. So we have availability, affordability, acceptability, and also accessibility. So this is 4A that we are focusing on PLANETIUM. And then we have now 1, 2, 3, 4, 5, 6, 7 of the research center inside the PLANETIUM, which is we have Sustain, Basics, SINA, Frozen Zoo, Earthlink, Spheres, and Nature. So soon at the level 4 of the Jalan Hospital campus, we're going to have a gallery of the PLANETIIUM. Okay, this is like a frozen zoo that I talked about much just now. And then we have Sina that working on people in special needs, okay, lead by Dr. Haliza. And then we have Earthling, lead by Dr. Hanif, that empower or distribute knowledge from our findings to the community. And then we have Sustain for Dr. Azri that try to find a new method or technology related to food security that uncommon, that extraordinary to ensure that the problem is solved. Okay, like using aquaculture that using soilless technology. Okay, we have basic. This is like to showcase the sustainable agriculture practice that we are improving, getting better in the 1 acre of the land that been given inside the IUM to us. Okay, and then lastly, I have one poem to end my talk just about in yesterday's week. We need to think about what happened yesterday to ensure that we can improve or be better for tomorrow, okay? Yesterday whispered secrets old and new, a canvas paints in every hue. Through trial, feast, and lessons learned, a brighter flame within us burned. Each moment shaped a path unknown, seeds of wisdom gently sown, echoes linger of step we tread, a dance of thought, the words unsaid. Yet in the past, the future gleams, a bridge of hope, a trails of dreams. For every tears and smile we share, builds the life we choose to bear. So thank you very much. Assalamu alaikum warahmatullahi wabarakatuh.
Waalaikum salam warahmatullahi wabarakatuh. May I kindly invite Prof to take the center stage? Thank you very much, Professor Lokman, for such an insightful and thought-provoking lecture. Let's give him another round of applause. Indeed, today's lecture reaffirms that Professor Lokman's contribution to science and planetary health are far-reaching, with the recognition and support of many collaborators in the field. To mark our appreciation, I would like to invite Professor Dr. Samsul bin Rahman, accompanied by Assistant Professor Dr. Rizwan Nul Hakim, to present a token of appreciation to our distinguished speaker. Thank you, Professor Samsul. Dr. Rizwan, please usher Prof to his seat. Thank you. Next, I would like to invite Assistant Professor Dr. Muhammad Sukri bin Baba, representing Office of the Academic Support and Services, to present the token of appreciations to Professor Lokman. Alhamdulillah. Thank you, Dr. Shukri. Once again, our heartfelt gratitude to our Professor Lokman for his time and dedication in sharing his expertise with us today. Alhamdulillah. Before we conclude our event today, we would like to take a group photo. We kindly ask everyone to stand and remain in their respective places. Okay, we would like to request everyone to come forward so that we can have a nice coverage of photo of the attendees today. Really appreciate your cooperation. Thank you. The exhibitors, if you can come forward as well, the exhibitors.